For the past year, you've probably heard the coronavirus thrown around often. However, coronaviruses are an entire family of viruses characterized by the spikes on their surfaces. The one that we've been focusing on is SARS-CoV-2, short for Severe Acute Respiratory Syndrome Coronavirus 2. This is the virus that causes the disease called COVID-19. Its first cases in humans were found in Wuhan, China, and thought to have originated from Chinese horseshoe bats, since it has a very similar genome to a coronavirus found in these bats. It could have been transferred to humans through contact with the bats or even through contact with another animal that got the virus from a bat first. SARS-CoV-2 is composed of a strand of RNA enclosed by nucleocapsid protein. This nucleocapsid is then located inside of a viral envelope made of lipids. It being an enclosed virus allows its RNA to enter the host cell via fusing its membrane without the host cell. SARS-CoV-2 was able to infect humans because it has surface proteins, called spike proteins, that are able to bind to ACE2 receptor proteins on the surfaces of human cells. The cells that SARS-CoV-2 infects are the ones with ACE2 receptor proteins, mostly found in the respiratory tract and intestines. After SARS-CoV-2 binds to the ACE2 receptor proteins and fuses its membrane with the host cells, it causes the host to make many copies of it, eventually leading to the cell lysing, which is also when the virus leaves the cell. This makes it a lytic virus. Right now, the CDC has acknowledged 3 major variants of SARS-CoV-2, but there are thousands more that we do not know as much about. Variants arise when mutations occur in the RNA of the virus. A coronavirus variant is also considered a strain if it has physically different features. There is B117 identified in the UK, which spreads faster and more easily and might even be more lethal. B1351 identified in South Africa, and P1 identified in Japan coming from travelers from Brazil, both change the shape of the virus, allowing it to better bind to ACE2 receptors and be affected by fewer of the established antibodies in people. The Pfizer and Moderna vaccines still work against these variants, but have shown to be somewhat less effective. In the future, the vaccines may need to be changed in order to keep up with the mutations. Speaking of vaccines, let's address how they work. Both the Pfizer and Moderna vaccines contain mRNA encased in lipid nanoparticles to allow it to be taken up by cells via endocytosis and prevent the mRNA from being broken down by enzymes in our bodies. When a vaccine is introduced, the mRNA is taken into a cell where ribosomes create the protein it codes for. In this case, one of the spikes found on the surface of SARS-CoV-2. Alone, one of the spikes is harmless. However, our immune system soon realizes that the spike protein does not belong on the cell. B and T cells are sent a response. B cells recognize the spikes as antigens and create antibodies to neutralize them. T cells can either be helper T or killer T cells. Killer T cells recognize infected cells and eliminate them, stopping further spread of the virus. Helper T cells can activate B cells to produce antibodies and T cells to kill target cells. After these cells are activated, a few become memory cells that stay in the body for much longer and are able to fight off the target if they encounter it again. This is great because if the host now contracts active SARS-CoV-2, the memory T and B cells will be able to react much faster because they recognize the spikes as something negative. Despite all the progress we've made with the vaccines so far, we're still not out of this neck of the woods yet. So, remember to wear your masks and wash your hands and stay safe.